Hello, hello, hello. Sorry for this short delay. Can you hear me or can you see me? Am I audible? Good evening, everyone. Okay, is it too light on my face? No. Okay. Uh, okay, now it's okay, no? Okay. Good evening, everyone. Yeah, I just got some confusion. So, yeah, how are you all? How are you all? Fine. Okay. I'm very good. Thank you for asking. I'm very good. I'm really good. Busy? What happened? Why are you too busy? Like you're preparing for the exam? Were you preparing for the exam? Oh, yeah. So you have studied everything, right, Abraham? Okay. Okay, so we will go to the next chapter. Last day we discussed about the first chapter, that was crop production and management. I hope everything is clear in that chapter, and we'll move to the next chapter, that is microorganisms, friend and foe. Actually, this is a very interesting chapter. We can relate all the things in this chapter with our everyday life. Everything in this chapter can be related to our everyday life. So it will be easy for you to study this chapter. Okay. So have you seen microorganisms? Have you ever seen microorganisms with your naked eye? With your naked eye? No, yeah. So what is a microorganism? So you cannot see the organisms with your naked eye. That is called microorganism. That can be only seen with the help of microscope. Okay, so microorganisms are those organisms which cannot be seen with our naked eye. Okay, so that are microorganisms, and there are few examples for microorganisms. Okay, so we will brush up this chapter only because it is actually a little bit lengthy chapter. So I don't think I can cover up all the things in this just 1 hour, but I'll try to cover everything. Okay, so microorganisms can be broadly categorized. Okay, can be broadly categorized into some groups. Okay, so they are, first is bacteria. Now, the next category would be fungi. The another category would be protozoa. Another category would be what? Can you give some examples? Can you give some examples? Yeah. Very good, very good. Algae, virus. Yes, algae. Yeah, virus, you're helping me. Yes. Good. Yes, algae virus. So these are some examples or some categories of microorganisms. Okay, so we will start with all of, or we'll just see what are some examples or some examples included in each of this. Okay, so first is bacteria, and the other is, you can see here, what is this? Do you know what is this organism? It's given in your textbook. Do you know what is this organism? Yes, algae. And the name of this algae is Chlamydomonas. Chlamydomonas. This is Chlamydomonas. And this one is Spirogera. Why it is called Spirogera? Why it is called Spirogera? What would be the reason? It is, yes, the cells. What is spiral actually here? What is spiral? It is spiral means it is meaning like this organism is spiral, but the organism is not spiral. No, actually, the chloroplast is spiral here. Okay, it has a ribbon-shaped chloroplast. It has a ribbon-shaped chloroplast. Okay, and that is why this organism is called Spirogera. Do you understand why this organism is called Spirogera? Are you clear? Yes. So, Chlamydomonas and Spirogera, they are examples of algae. And some other category of microorganisms are protozoa. Can you say what is these organisms here? I'm facing some issue with my mouse, so I'm a bit confused with this my mouse that I'm using here. Okay, so yes, very good, very good. I'm getting the answers. This is amoeba, and the next one is paramecium. Okay, paramecium. Very good, very good. Smart. Yes. So these are some examples of what? Yes, protozoa. So protozoa is another classification of microorganism. And yet another classification of microorganisms are fungi. So what is the speciality of fungi? What is the speciality of fungi? It produces it actually produces spores for their reproduction purposes. Okay, so they actually produces spores, and this is also a category of microorganisms. And what are these organisms that you can see here? Yes, very good. Bread mold. Why it is called bread mold? Why it is called bread mold? Why it is called bread mold? Because these are grown on the bread that we eat. Okay, these breads are contaminated. It is actually not present in bread. It can be, yeah, it is grown on bread. Okay, so due to some unfavorable, due to the favorable conditions that they need, they grow on bread. That is why it is called bread mold. So how can you see this bread mold on bread? Like we can see some black colored or dark colored spores or some powdered substance when we keep a bread for some long time. Okay, so that is bread mold. That is rhizopus, and the scientific name is rhizopus. Okay, this is bread mold. And what is this organism? Patches. Yes, very good, very good. What is this organism? I'm waiting for the answers. This is penicillium. Okay, this is, yeah, this aspergillus and penicillium actually, they look similar, right? So it is a bit confusing. Okay, so this is penicillium and this is aspergillus. Okay, so what is the speciality of penicillium? What is the speciality of penicillium? Have you ever heard of the antibiotic penicillin? Have you ever heard of the antibiotic penicillin? Actually, the antibiotic penicillin is derived, or it is formed from this microorganism, Penicillium notatum. Okay, the first antibiotic was penicillin, and it was derived from Penicillium notatum. So that we will see later. Okay, so rhizomes or rhizobium, what? I didn't get it. Which one? Rhizobium is another bacteria. Okay, rhizome is a structure that is seen instead of roots. It is not seen everywhere. Okay, in every plants. Okay, so rhizobium actually is a soil-borne bacteria. Okay, but these are not rhizobium. Rhizobium we will study in another chapter in higher grades. Okay. Okay, Roshan. Okay. Yeah. Yes. Okay. So these penicillin, the antibiotic penicillin is derived from Penicillium notatum. And this is another organism or microorganism, that is aspergillus. So these all, Rhizopus, Penicillium notatum and aspergillus, they all comes under which group? Fungi. Okay. So these comes under fungi. And next, yet another classification of microorganisms is viruses. So what is the speciality of viruses? Do you know? What is the specialty of viruses? What is the specialty of viruses? Can they live on their own, or can they survive on their own, or do they need a help? Okay, yeah, reproduces in host cell. They actually need a help. They cannot live without a host, right? So, uh, like you all have best friends, right? So, like a thick best friend you will have. So you always say, "I cannot live without you," or a lover to a lover, you will say, "I cannot live without you," right? So that is the thing here. Okay, these viruses cannot live without a host. Okay, they need a host cell. That may be a bacteria, or that may be a plant, or that may be an animal. Okay, so they cannot live without a host. They reproduces only inside the host cell. Okay, so they cannot survive without a host cell. They cannot even survive. Okay, that is a specialty of what viruses. So the shown viruses here are, this is a Devabala. Oh, that's okay. That's okay. We have just started only, Devabala. Okay, we have just started only. So these are five categories of microorganisms that I am teaching now. First is, these are just some examples. Okay, so we have not gone so far. Okay, so you can hear from this. So this is bacteriophage, and this is what? This is another type of virus. There are different types of viruses, but you can specifically say this is a bacteriophage. This is a type of virus. Okay, so so these are the five categories of microorganisms. Can you please just say what are the five categories of microorganisms in the chat box? Can you please list out? Yeah. Yeah, everyone is giving me the answers. Yes, correct. Yes. Very good, very good. You have already studied. Yes. So, bacteria, fungi, algae, protozoa, and viruses. So, these are the five categories of microorganisms. Is it clear? Everyone, is it clear? Very good, very good. Yes, yes. Okay. So we will move on to the next point. Okay. So we can just discuss some questions here. Classify bacteria. Actually, the questions I have put a lot of questions in this class, because it is your exam, right? So this is your revision class. So you have to more focus on the questions part. Okay, so concentrate on this. Classify bacteria on the basis on their shapes. Actually, this is not specifically given in the textbook, but this can be asked. Okay, this can be asked because it is a general question. Okay, so classify bacteria on the basis of their shapes. Do you have any idea on this? On the based on their shape, how bacteria can be classified? Any idea? Anyone? Anyone? Okay, that's okay. Yeah. Okay, I'll tell you. So these are different shapes on in the category of bacteria that we can see. Okay, one is cocci. Cocci means it is spherical in shape. Okay, spherical in shape. The other one is, what does it look like? It looks like a coma, right? It looks like a coma. Can you relate this with a coma? So this is a coma-shaped bacteria, which is generally called Vibrio. And this is a rod-shaped bacteria. Okay, this is rod-shaped bacteria called Bacillus. And these two categories, spirochetes, spirilla, okay, and some other ones are also there. So they can be said as spirillum, that is spiral shape. Okay, so these are the four basic categories of bacteria based on their shapes. Okay, so coccus or cocci, then vibrios or vibrio, then third one is bacilli or bacillus, and the fourth one is spirilla. Is it clear? Everyone, is it clear? Yeah, my students are very active, actively responding to all my questions. Very good, very good. Yeah. So these are the different categories of bacteria based on their shapes. Ah, yeah. Yes, very good. There was a question in my paper. Yeah, someone have given me the comment box. There was a question in my exam question paper. The rod-shaped bacteria are called. What was the rod-shaped bacteria? Thank you, Bhavna. Bhavya. Yeah, Bhavna. Bhavya. Yeah. So what was the rod-shaped bacteria called? Here itself you can see. Yes, very good. The answer is Bacillus, right? So just you can remember these shapes. Sometimes questions can be asked because like we think it is not there in textbook, so it can't be like it won't be asked in the answer question. But just remember these general things, okay? So that you can answer, you can score there. Okay. So rod-shaped bacteria are called Bacillus. Spherical-shaped bacteria are called cocci. Coma-shaped bacteria are called vibrios, and spiral-shaped bacteria are called spirillum. Okay. Now, yeah, how are viruses different from other microorganisms? How are viruses different from other microorganisms? How? I have already said that. Yeah, other microorganisms like bacteria, fungi, algae, protozoa, actually they all doesn't need a host to survive, or they all doesn't need a host to reproduce, okay, for the next generation. But in case of virus, they actually need a host or like bacteria. It can be bacteria, okay, that host organism can be bacteria, it can be a plant, or it can be an animal. Yeah, I'm getting the answers. Virus reproduce inside the cells of the host. Very good. Okay, so this is the main feature of virus that differentiates them from other microorganisms. Okay, so we will see the answer. They are too much smaller in comparison to other organisms. Yes, it is also correct. In comparison to fungi, like we can see some fungus with our naked eye. Can you see one example? We can see like one type of fungus with our naked eyes. Not only one, but can you see some examples? Fungus that can be seen with a naked eye. Bread mold, actually, can you see the structure of bread mold? It is not visible, right? We can observe the bread mold as a black powder only. In order to see the structure of this bread mold, we have to use microscope, right? So it cannot be seen with a naked eye. But is there any other examples of fungi that can be seen with a naked eye? Is it? Is there? Yeah. Lichens, actually, it cannot be given as an example of a fungus. Okay, lichen is actually not a fungus. It is a combination of a fungus and an algae. Okay, it cannot be considered under this fungus. Penicillium. Actually, the structure of penicillium is also, we can see only through a microscope, right? Have you seen mushrooms? Mushrooms. Can you think of mushrooms? Mushrooms is actually a fungus. Okay, so these mushrooms, do we need a microscope to see mushrooms? No, right? No, yeah. So mushrooms is a fungus that can be seen with the naked eye. Okay. So there are some other examples of other microorganisms that can be seen with the naked eye. But these viruses, they are very small enough, very, very, very small, that we cannot see with our naked eye. And also it in the size, it is also much smaller in comparison to other organisms. Poisonous mushrooms are colorful. Yeah, very good thought or very good fact that we can share with everyone. So Abraham told us poisonous mushrooms are colorful. Okay, so be aware of these colorful mushrooms, vibrant colored mushrooms. So they are poisonous. Okay. So viruses cannot survive without a host. So that is the main important point that helps viruses to differ from other microorganisms. Viruses cannot survive without a host. They reproduce only inside the cells of the host, and that may be bacteria, plant, or animal. Is it clear? Can I go to next one? Is it clear, everyone? Yes. Okay. Yes. Okay. Now, bread mold is an example of I want the answer from everyone. Yes, it is fungus. What is the scientific name of bread mold? Can you say? What is the scientific name of bread mold? You can just think of that. Or you can just memorize the scientific name also. Not rhizobium, it is rhizopus. Okay, rhizobium is a bacteria. Okay, rhizobium is actually soil-borne bacteria. Okay, that can be seen in a leguminous plants like a pea plant. You have, pea plant, right? So in the roots of these leguminous plants, we can see rhizobium. So that is not a fungus, that is a bacteria. Okay, so bread mold is rhizopus. Don't confuse that with, okay? Okay. Yes. Now, give two examples each for bacterial diseases and protozoal diseases. These can be memorized. Like these can be memorized only. So this cannot be taught to everyone. You can remember this or you can memorize these things. Okay. So it is given in your textbook. That is some bacterial diseases. Yeah, very good. Everyone. Yes, everyone. I'm getting the answers from everyone. So dysentery is an example of a protozoan disease. Okay, and it is caused by causative organisms. You need not study, but you must know the type of causative organism. Okay, it should be known to everyone the type of causative organism. So dysentery is caused by protozoa. Malaria is caused by protozoa. You can remember this also, na? Plasmodium. You can remember this also. Okay, Plasmodium is the protozoa that causes malaria. And typhoid. Typhoid is a bacteria, bacterial disease. Okay, I just forgot the word. Typhoid is a bacterial disease. Tuberculosis is also a bacterial disease. So these are some examples. You can recall some other examples of these bacterial diseases and protozoan diseases. Okay, so these are some examples. Now, the next one is, there are so many living things nearby that we typically cannot see with our naked eye. One of the types is given below. Based on this, answer the following question. Oh, I have not even read the question. I'm getting the answers. Anyway, you guys are very smart. Okay, very smart. Okay, so identify the type of microorganisms shown above. What is the type of microorganisms? Very good. Can you specifically say what is this microorganism? Yeah, it is a virus. And can you specifically say what is this? Can you specifically say this is a virus called coronavirus? Okay, that is the answer of third one. Okay. Yeah, I got it. So bacteriophage. Okay, so this is a virus. And if you need, you can write bacteriophage also. Okay. Okay, if you write bacteriophage also, you'll get more smarter than other students. Okay, so where do they reproduce? Where do they reproduce? They reproduce in host cell. That may be a bacteria, that may be plant, or that may be animal. Okay, give two examples of diseases caused by them. So viral diseases, you can give some examples of viral diseases. I know you all know this disease is caused by virus. Yes, influenza or flu. Yeah, influenza or flu. Then the common cold. You always get cold, right? So this is caused by rhinovirus. Okay, so common cold. Then chickenpox. Chickenpox. How many of you have already gone through chickenpox? I have already got chickenpox when I was in 10th standard, and it was very hectic. I didn't get. Okay. So be aware. Okay. Yeah. So, so these are some examples of diseases caused by them. Okay. Common cold, influenza or flu, polio, chickenpox. I got COVID instead. Yeah, I also got COVID 2 times. Yeah. So, actually, there are. Yes, I also got COVID. Yeah, everyone is getting COVID. Like I also got COVID when I was when I was in another place, and after when I that was hectic, and when I reached home, I got another set of like another round of COVID. Yeah. So yeah, okay. So I just got yeah. So yes, I am getting the answers. So there is another doubt here, ma'am. Is hepatitis A viral disease or not? Actually, hepatitis A is a viral disease. Okay. Yeah, sometimes the questions can be asked. Hepatitis A is a viral disease. Okay. So now we can move on to the next category, next topic, that is friendly microorganisms. Actually, microorganisms can be both friendly as well as harmful. Okay. Yes. Yeah. Okay. So microorganisms can be friendly as well as they can be harmful also. So be aware of microorganisms. They will be friends. Okay. They will be friends, but they will be enemies for us too. Okay. So we all use or we all drink milk on a daily basis, right? So do you know how this how a curd is made? Have you ever seen your mother putting some spoon of like a few part of curd into the milk, and the next day it is that whole milk is transformed into curd? Yeah. So what is the action taking place there? What is the action taking place there? Actually, we know that this milk, okay, milk, it contains lactose sugar. Okay, milk, it contains lactose sugar, and the curd has lactobacillus bacteria also there. Okay, the curd has lactobacillus bacteria. So this lactobacillus bacteria, it converts lactose into lactic acid. Okay, it converts lactose into lactic acid. Okay, so what happens here? Lactic acid is actually an acid, right? So what happens there? It coagulates the milk. It coagulates the milk protein. And that milk protein is actually called casein, that you don't want to study here. Okay, so the acid environment coagulates the milk, and that milk is converted into curd. Okay, do you get it? Do you get it? Do you want me to explain it once more? Everyone, is it clear? Can I move to next one? Yes. Okay. So milk is converted into curd with the help of, okay. I will explain once more. We know that milk contains the sugar called lactose. Okay, lactose is called milk sugar. You have already studied lactose is milk sugar, right? So lactobacillus bacteria will convert this lactose into lactic acid. Okay, so what is the specialty of this lactic acid? Lactose itself is not very it does not make a milk into curd. Okay, so this lactic acid, okay, this lactic acid is an acid. Okay, it coagulates the milk protein, that is called casein. It coagulates the milk protein called casein, and that results in the formation of curd. Do you get it? Do you get it, Devabala? Is it clear? Is it clear? Yes. Okay. Yes. Okay. So I will move on to the next one. Okay. So have you tried making bread at home? I have not tried, but this is a process. Okay. Oh, yes. Very good. Akshat, have you tried? Very good. Yeah. So how this process takes place, how the rising of dough of bread takes place. Okay. So the main ingredients we use here are water, yeast, sugar. Yeah. And the flour. Okay. So the ingredient that helps in the rising of dough is yeast. Yeast is a microorganism. This is a fungus. Okay. Yeast is a fungus. So what happens here is that yeast in the presence of sugar. Okay. Yeast in the presence of sugar. We also add sugar here, right? So yeast in the presence of sugar undergoes anaerobic fermentation. Sorry, anaerobic respiration, or it is also called fermentation. What is it? Without the presence of oxygen. Without oxygen, they will respire. Okay. So yeast respire anaerobically, and carbon dioxide and alcohol will be formed. And what happens here is that this carbon dioxide results in the rising of the dough. Okay. This carbon dioxide helps in the rising of dough. Carbon dioxide is a gas, right? So it actually results in the rising of dough. Do you get it? Did you get everything? Like everyone, is it clear for you, everyone? Yeah, I'm getting thumbs up. Yes. Okay. So I'll move to the next one. Okay, we have a limited time actually. So I'll move a little faster. So this is a reaction taking place. Sugar plus yeast. Sugar plus yeast in the absence of oxygen will give out carbon dioxide. And alcohol. Okay. Oh, actually, my net is actually stable. I don't know why. Okay. I'll make it more clear to you. Okay. Is it audible now, Isabel? Is it now audible? Is it clear? My voice is clear? Yes. Okay. Okay. Thank you. Now, this anaerobic respiration is called fermentation. Now, what is this? Friendly microorganisms. First, we already saw about this household products, and this in case of medicines, medical field. Okay. Medicinal products. These are antibiotics. So I have already said what is an antibiotic. Yeah. So the first antibiotic discovered was penicillium. Sorry, penicillin, right? And it was derived from Penicillium notatum. Okay. So how was it discovered? Who discovered that? This also you should study. Okay. Alexander Fleming discovered the antibiotic penicillin. So how was it discovered? Okay. There's a story behind that. Actually, Alexander Fleming was studying on some other bacteria called Staphylococcus. Okay. He was studying on Staphylococcus bacteria, and he had a culture plate with this Staphylococcus bacteria. Okay. And he left this culture plate with Staphylococcus bacteria in the laboratory, and he went for a vacation. He forgot to cover that. Okay, he went for a vacation, and after 2 weeks, when he returned back, he saw some other fungus. Okay, some other organism that was like flourishing in that same culture plate, and that was discovered as penicillin. Sorry, Penicillium notatum. Okay, that fungus was Penicillium notatum. Okay, and what was the specialty of this Penicillium notatum? Okay, what was the specialty of this? And the specialty of this fungus was that it inhibited the growth of Staphylococcus. It inhibited the growth of Staphylococcus. So that was how it was actually an accidental discovery. It was not a non-discovery. It was an accidental discovery. Okay, so this is the history behind the discovery of the antibiotic penicillin. Is it clear? Sometimes it can be asked. The story can also be asked in your question paper. Okay, is it clear? Okay, yes. Okay, I'll move to the next one. So these are the specialities of antibiotic. Voice break. Okay. Sometimes I don't know, the network is, sometimes it will be unstable. I don't know. For everyone, is it clear? Is my voice breaking now? It is clear. Okay. Okay, so check your network, check yours network connection also. Okay, I think my network connection is stable. Okay. Okay. Yes. Now, these are some peculiarities of antibiotics. Okay, when this antibiotic is given to our body, it actually inhibit the proteins. It actually inhibit the protein synthesis of bacteria that were that like that attacked us. Okay, it inhibit the protein synthesis. It inhibit the nucleic acid synthesis. Inhibit the cell wall synthesis. Disruption of cell membrane function. What is like left for the disruption, right? So that the bacteria, the whole bacteria will be erased from our body. Okay. Yes, tell me, Roshan. Tell me. So this is the definition of antibiotic. Antibiotics are medicines that fight Antibiotics are medicines that fight bacterial infections in people and animals. Okay, so they are medicines. When we get a fever, okay, when we get a cold, we run to the doctor and the doctor will prescribe us some antibiotics, right? So what is the function of this antibiotic? These are medicines that fight the bacterial infection. Okay, these fight the bacterial infection in people and animals. Okay, less than the first one. Yes, really, because actually when the bacteria enters our body itself produces some antibodies. Okay, our body in order to fight that bacteria. So that antibodies will be that remains in our body itself, and that when that bacteria again attack that for the second time, we have this immunity power. So that is why the second time that illness will be less than the first one. Okay, Roshan, did you get it? Get it? Yes, okay. So here, the next useful use of microorganisms in this is in the production of vaccines. Yeah, every one of you might have taken this coronavirus vaccine, right? On that that period where coronavirus attacked us, right? So what is the function of vaccination? That was like discussed just now. What is the function of vaccination that is given to us? Okay, vaccination is the injection of a dead or weakened organism that forms immunity against that organism itself. Okay, so in the body. So what is vaccination? Actually, this vaccination to kill the bacteria, very good. To kill the foreign particle. Okay, so actually, what is vaccine? Vaccine actually contains a killed, suppose this is a microbe. Okay, suppose this is a microbe. And I need to produce vaccine against this microbe. So I'll take a part of this. Okay, or that part is called antigen. Okay, I'll take a part of this, or I will kill that one, or I will weaken its effect. I will weaken its effect. And I will introduce this as vaccine to my body. Okay, to my body. Okay, what happens in my body? What happens as a result of this in my body? My body produces antibodies against this antigen, right? Because this is a foreign particle. Okay, this is a foreign particle. Okay, my body produces antibodies against this antigen. Okay, now this antibodies remains in our body. And the second time, the same bacteria or the same microbe, when it enters our body for the second time, what happens? These antibodies will go and attack them. Go and attack them. Okay. So this is the same principle in vaccination. It is an injection of a dead or weakened microorganism that forms immunity. And that power is called immunity against that organism. Did you get it? Everyone, is it clear? So this is also called immunization. So immunization is a process by which an animal or a person stays protected from diseases. Okay. I'm going little more faster. Okay. Antigen is actually a small portion of antibody. Of the microorganisms. I'm really sorry. So antigen is the small portion of the microorganisms or the bacteria, any foreign particle that enter our body, that any part of that foreign body is called antigen. In detail, you will study later. Okay. This is just your 8th standard. 9th standard, there are another chapters coming on. You will study more in detail. Okay. Now, the next advantage of microorganisms is that to increase soil fertility. To increase soil fertility. So how does it increase the soil fertility? With the help of some biological nitrogen fixers. So bacteria that are able to fix nitrogen from the atmosphere to enrich soil with nitrogen and increases it, bacteria is called biological nitrogen fixers. Okay. So yeah, here rhizobium bacteria. This is also an example. Okay. Can you give some other examples? Some other examples. Can you see here? Cyanobacteria or blue-green algae. It is also acetobacter. Yes, very good, very good. Yes. So these are some examples of biological nitrogen fixers. Now, name some commonly known antibiotic. Actually, my mouse is not working. That is why I'm getting a little lag. Okay. So, name some commonly known antibiotics which are made from fungi or bacteria. Some commonly known antibiotics. Just one you can say now. Right? Yeah, penicillin, very good. From fungi, penicillin is obtained. Cephalosporin is another example. From bacteria, streptomycin, erythromycin, tetracycline, chloramphenicol. So these are some other examples of antibiotics derived from or which are made from bacteria. Okay, so this all things you have to memorize it. Okay, you have to memorize it. That is a task in this chapter. Okay, so what will happen if we take antibiotics when not needed? When not needed, okay, what will happen if we take antibiotics when not needed? What will happen? If you are taking continuously an antibiotic, what will happen? Resistance will be there. Okay, resistance. It also kills the beneficial organisms, beneficial microorganisms in our body. Okay, that is a side effect of this antibiotic when we are taking this antibiotic continuously. So don't ever take antibiotics with a prescription, without the prescription of a doctor. Okay, he only knows at what dose you should take that. Okay, so do not take antibiotics when not needed. Okay. Yeah, so bacterial resistance or antibiotic resistance will develop. So what is antibiotic resistance? It may make the drug less effective when you might need it in future. And it also may kill the beneficial bacteria in the body. Okay, so I'm going to the next one. Now, explain the discovery of penicillin. Yeah, it is a story. Actually, it was asked in some other year. Okay, explain the discovery of penicillin. You all know, I have already said how the penicillin was discovered. It was an accidental discovery. Okay, so who discovered penicillin? Who discovered penicillin? Who discovered penicillin? Alexander Fleming. Resistance means like inhibition. That is resistance. Okay, someone have asked me what is resistance. Actually, it can be simply said as an inhibition. Okay, something we don't like or something which is inhibited. That is the meaning of resistance. Okay, is it clear? Yes. Okay, so actually, this is the discovery of penicillin. This is the original experiment which was done by Fleming. And here you can see the penicillium colony. And this is Staphylococcus bacteria. Towards this side, where penicillium is not there, there are big colonies of Staphylococcus bacteria. But when coming closer to this penicillium colony, you can see it undergoes lysis. That means the colony becomes much and much smaller, or it have undergone lysis because there is a zone of inhibition here. Okay, this is a modern experiment, and here you can see this is penicillium and this is Staphylococcus. You can see a zone of inhibition here where Staphylococcus cannot grow. Okay, so this is the experiment. Okay, now alcohol is produced in a massive scale with the help of microbes, right? The microorganism that helps in the production of alcohol. Very good, very good. It is yeast. It is yeast. Now, mention the natural sources from which the microbe is taken. Some natural sources. Some natural sources are wheat, rice, barley, etc. Okay, you can study some examples also. Now, define the process that takes place in alcohol production. Yes, what is the process? Yeast reacts with the sugar in the absence of oxygen to give carbon dioxide and alcohol. Okay, everyone, is it clear? Yes, define the process. Fermentation. What is fermentation? You all know what is fermentation, right? It is the anaerobic respiration that takes place with the help of yeast. Okay. Now, match the following. You can match the following. What is blue-green algae can be matched with, to which it can be matched? Blue-green algae. Yeah, very good. Now, streptomycin. It is an. Yes, I want the answer. 3. Very good. Yeah. Lactobacillus helps in. Very good. Curd formation. And Edward Jenner. Edward Jenner. Very good. Vaccine. Actually, Edward Jenner is a scientist who discovered vaccine. And that vaccine was for smallpox virus. Okay. It's for smallpox. Just remember that also. Okay. Yes. Now we will move on to the next one. Sorry. Yes. Why do idli and dosa maker add some yeast or all batter of previous, not previous day. Okay. It is previous day. Previous. Previous day to the newly made batter and keep it for 1 day. For the dough to rise, what is the reaction? Which gas helps in the rising of dough? Which gas helps in the rising of dough? I want the answer. Very good, Adit. Very good, Amin. Sorry, Amreen. And yeah, everyone. So carbon dioxide. Very good, Bhavya. Now, how milk is turned into curd? With the help of, yeah, you know the process, the whole process you should know. So which bacteria help in the process of turning this milk into curd? With the help of lactobacillus. Very good. So milk is converted into curd by the process of fermentation. It is converted into curd by the help of bacteria. Lactobacillus convert lactose present in the milk to lactic acid. And the lactic acid coagulate the milk protein casein and results in the formation of curd. Now, it is actually not moving. Yeah, okay. Now, this, so we have already said what a friendly microorganisms are. We have given, like, we have seen so many examples of some friendly microorganisms. And here are some microorganisms that are very harmful to us. Okay, that causes diseases. Harmful microorganisms are those microorganisms that causes diseases in us, plants and animals. Okay, yes. So you have to memorize this. Okay, this is another thing to memorize. This is another task for you to memorize. Okay, so some disease causing microbes in human. Bacteria. The bacteria causes tuberculosis in humans. Okay, tuberculosis, cholera and typhoid, and virus. In case of virus causes measles, chickenpox, polio, hepatitis A. And these are given this mode of transmission. Okay, through air. Tuberculosis is spread through air. Cholera is spread through food or water. Then measles through air. Chickenpox through air or contact. Sorry. Now, polio is spread through air or water. Hepatitis A is spread through water. Now, protozoa. Okay, protozoa. Example is malaria. So, malaria is spread through mosquito. Actually, to be more specific, female Anopheles mosquito. To be more specific, it is female Anopheles mosquito. Okay, remember that thing. This is very important. Okay, now this is causing microbes in plants. Bacteria causes citrus canker and it is transmitted to air. Then virus, it causes some yellow vein mosaic of bindi. What is bindi? What is bindi? It is actually Hindi word. What is windy? Or crab? Yeah, it is lady's finger. Yeah, it is simply called lady's finger. Okay. So it is transmitted through insects and some fungal diseases or rust of wheat. It is transmitted through air, seed, etc. And some disease causing microbes in animals. Okay. So bacteria causes anthrax disease in both humans and also in cattle, and some virus cause foot and mouth disease in cattle. Okay. So these are some examples of harmful microorganisms. Just memorize it, study it, and like it can be asked for 1 mark. Okay. Can be asked for 2 mark. Some small uh like marks it can be asked. Okay. Study this. Now, how can we prevent the spread of communicable diseases? How can we prevent the spread of communicable diseases? Actually, what is communicable diseases? Communicable, it is also already there in the question. Communicable diseases are those diseases that can be spread from one person to another. Okay, if this person has a communicable disease, okay, if this person has cold, okay, when he sneezes, no, the second person the second person near to him, sometimes the next day he will also get cold. Why? It is spread through the droplets. The droplets. Okay. Yeah. Now, how can we prevent this spread of communicable diseases? Yeah, I'm getting too many answers. Yes, very good. By not using contaminated food is very important. By avoiding physical contact, yes, just just stay some isolated with this person having communicable disease and spreading an infected. Yeah, by not going near when they are coughing or sneezing is very good. Also, vaccine. Yeah, the most important points I got it from this chat box itself. Okay, wear mask. Yeah, we already faced some corona period, right? So all of us wore this mask every day on a daily basis, right? So what was the purpose of that? To prevent the spread. Okay, so good hygiene, protective equipment like mask, vaccination, and clean water. You must drink clean water. Okay, wear mask. I still wear mask daily. Very good. Okay, very good, Akshat. Very good. It should be done. Okay, very good. Eat clean food and water. Yes. Now, given below a few diseases: tuberculosis, measles, chickenpox, polio. So select the odd one out with respect to the causative microorganisms. Which is the odd one out here? With respect to the causative microorganism, okay, the type of microorganisms causing these diseases. I'm getting different responses actually. Yes, one said it right. That is tuberculosis, measles, chickenpox, polio. Okay, they all are actually what? They all are actually caused by virus. But tuberculosis is caused by bacteria, right? Yeah. So tuberculosis is the odd one out, right? In a preventive measure for the given disease. Yeah. Give me. Keep the patients in complete isolation, vaccination. These are some common methods to prevent this disease, right? Yes, vaccination is a very important thing. Okay. Now, coming to fruit preservation, we all have seen our parents putting some pickles, mangoes in salt water. Okay, so these are some food preservation methods. Okay, how the food gets spoiled? How the food gets spoiled? In your life, in one way or the other, you all have gone through this food poisoning. You have gone to hospitals, you got some medications and done that, right? Like recently I got some food poisoning. Okay, so why this food poisoning is caused? How this food poisoning is caused? This foods sometimes can be attacked by microorganisms. Okay, and these microorganisms produces toxins that causes health issues or food poisoning in us. Okay, so this food can be preserved. Okay, to various methods. So these are some of the various methods involved in food preservation. Chemical method, benzoates. Okay, so there are few examples of this chemical method. Preservation by common salt, like mangoes, putting in common salt. Then preservation by sugar, preservation by oil and vinegar, by, like, for example, while making pickles. Heat and cold treatments. What is a heat treatment? Can you give an example of heat treatment? Heat treatment. Freezing is cold treatment. Okay. Heat treatment. Yeah, freezing, like we put everything in our refrigerator, right? In fridge to avoid the spoilage. But heat treatment. Yes, very good, very good. Pasteurization. What is pasteurization? What is pasteurization? We will boil the milk, not we, the food product, sorry, the milk producing companies actually, they actually boil the milk to extreme temperatures, 70 degrees Celsius. That is extreme temperatures, right? So, and for 15 to 30 minutes, and it is suddenly chilled and or cooled and stored, so that it prevents the attack of microbes. This is pasteurization. It is a heat treatment. Now, storage and packing. We always put the vegetables, some food in airtight containers or airtight packets, right? To avoid the entry of air. Okay, to avoid the growth of microorganisms. So this is storage and packaging. So these are some food preservation methods. Okay. Yeah, here you can see a lot of food preservation methods. This is refrigerator, this is wine, and yeah, this is freezing, milk, torn milk, fishes, everything. These are some examples of food preservation methods. Okay. Now, why do we keep food in a refrigerator? Because refrigerator already have a low temperature, right? Refrigerator already have a low temperature. Refrigerator already have a low temperature. So this low temperature inhibits the growth of microorganisms. Okay. So that is why we keep food in refrigerator. Now, a bread was kept by Rahul for days till he noticed some black fungus on it. His mother told him that the bread is poison. How is food poisoning caused? How is food? Sorry. Yeah, I have for pasteurization, I told 15 to 30 minutes, right? It is not 15 to 30 minutes. It's 15 to 30 seconds. Okay. Okay. So how is food poisoning caused? How is food poisoning caused? Microorganisms produces toxins. That is how food poisoning is caused. Mention any two methods by which food can be preserved. There are many methods: chemical methods, oil and vinegars, common salt, freezing, pasteurization, storing, packing, everything you can write. Okay. Now, we know that pasteurized milk can be consumed without boiling. Define pasteurization. You already know what is pasteurization. Who discovered pasteurization? Who discovered pasteurization? Who discovered pasteurization? Louis Pasteur. Okay. Remember this one. Louis Pasteur discovered pasteurization. Give the temperature required for pasteurization: 70 degrees Celsius. Okay. Now, explain nitrogen cycle. Yeah, we have come to the last session. Just 5 more minutes. Okay. Explain nitrogen cycle and draw a schematic diagram of nitrogen cycle. So what is a nitrogen cycle? We know that our atmosphere, the air we breathe, okay, it actually contains 78% of what? Nitrogen. Okay, the most abundant gas in atmosphere is nitrogen only. Okay, so when we look to the surface, actually, or when we look to the atmosphere, there are plenty of nitrogen present, but we cannot take this nitrogen as such. Okay, we cannot take this nitrogen as such. So there are various cycles altogether being processed in the atmosphere. That biogeochemical cycle is called nitrogen cycle. Okay, that cycle is called nitrogen cycle. So this is a biogeochemical process which transforms the inert nitrogen present in the atmosphere to a more usable form for living organisms. This Inert nitrogen, actually, it cannot be taken as such for us. Okay, or for the living organisms. It should be converted into a usable form. So this process is called nitrogen cycle. Okay, yeah, very good. I'm getting the answers. Now, so this is the nitrogen cycle. This image is already given in your textbook. So, it's in a simple form like N2. It is N2. Okay. Do you get it, Devbala? Do you get it? Okay. So this atmospheric nitrogen, actually the bacteria present in the soil, okay, this bacteria fixes the nitrogen present in the atmosphere, and this nitrogen is converted into nitrogenous compounds like nitrates or nitrites. Okay, actually, you don't have to study this in detail, but you have to know, you should know what is a nitrogen cycle and just memorize this pathway. Okay, memorize this pathway. And the compounds of nitrogen, so we got this compounds of nitrogen here in the soil, and the bacteria turn these compounds of nitrogen into gaseous nitrogen. It is reversed back to the atmosphere. Okay. Okay. So now this is one method of cycle. Okay. Another method with which this atmospheric nitrogen can be fixed is by lightning. It is a physical method. Okay. This lightning also fixes atmospheric nitrogen and it converts it into compounds of nitrogen in soil. And again, this bacteria turn it into nitrogen, gaseous nitrogen. Okay. And this compounds of nitrogen, how it is given to, or how it is taken by plants and animals. We know that the soil, plants grow on soil, right? So plants already get this nitrogen through soil. Okay. So plants will uptake this, and these plants are used by animals, like including us also, right? So it is this animals eat plants, and nitrogenous waste, we also provide this nitrogenous waste from excretion right after death also, and also from through death we provide nitrogenous waste are being given to the soil. So this nitrogen waste from excretion and death, and it is again returned back to the soil. So this is how this cycle operates. Okay, this as a short cycle, you should remember this cycle. Sometimes it can be given for a long answer question. So for that, you have to just draw this flowchart and explain this flowchart once. Okay. So these are the things that that is being covered in this chapter. Is there any queries? I know I have taken a little extra time, 5 minutes more for this session, but this is actually a very long chapter. I hope this session was very helpful for you. In our exam periodic test, yes, it can be asked. Nitrogen cycle can be asked. Okay, clear. Okay, thank you so much. So we will see with yet another chapter, conservation of plants and animals, in our next class. Okay, so take care all of you. Study well for the exam. Okay, study very well for the exam. So have a great night. Sleep well. Okay. Thank you so much. You can leave. Bye. Bye-bye. Thank you. Thank you. Thank you so much. Oh, thank you so much, Devala. Thank you so much. Have a good sleep. Bye.